On the last video, we talked about the structure of a gene, and we talked about how genes actually have non-coding regions, which include the promoter and termination sequences. The promoter sequence is the area that actually has to attach to those transcription factors in order to activate the gene for transcription. And it includes things like the data box, which responds to transcription factors and sends a signal for RNA polymerase, please copy here. Then, with the help of transcription factors, RNA polymerase will initiate the transcription process and keep coding downstream from that point the entire transcription unit until it gets to the termination site, which sends the signal that it's time to release and that RNA is completed. The actual RNA transcript, though, includes a lot of things, including a cap that produces energy and protection for the RNA message, as well as an upstream uncoding or untranslated region, which helps protect the message from degradation. And the longer that region is, the longer the RNA is going to survive in the cytoplasm. Then you get to the actual coding sequence, which includes introns and exons, and we'll talk about that in this video, what's the deal with those introns. Then you get to the stop codon. That means that the protein is done, but after that you still have another downstream untranslated region to protect the DNA code from degradation, and to add insult to injury, you put another poly A tail or a bunch of little A's, adenines, strung together to actually help protect the message from degradation. And that's how the transcription unit looks like when it's done after transcription in eukaryotic DNA. But in eukaryotic DNA, this needs to be processed because of those introns. Now, it's kind of hard to understand why they're there in the first place if they're not going to be part of the protein synthesis. Why does the coding sequence include a bunch of introns and exons, as you see in the bottom here? Well, the first thing you need to understand is what happens to those things. The exons are the actual pieces that actually code for the proteins. They include the codons, which are going to become part of the code that determines the protein sequence. In other words, this is all you want. You only want the exons, like it shows in the picture on the top. You want to remove all the introns so that the RNA will have only the pieces which are important for the protein synthesis. So why the hell did you put the introns there in the first place if you're just going to remove them? The exons are the express parts. The introns are the junk in between. But why would you leave them there? Now, at the beginning and the end of each intron, there's always something that's called an intron boundary sequence, or a signal that tells the cell, look, I'm junk. I'm just here because I have a role, but make sure that you take me out before you actually try to make the protein, because I don't want to actually be translated. I'm just here to do some other things, which we're going to talk about in a second. So they actually send a signal to the cell. Basically, let's say, for example, it's AAATGZ. Whatever that sequence is, will actually represent a piece that will tell the machinery of the cell that takes them out that this is the beginning and the end of the intron. Now, the machinery that actually takes that out, it's called the spliceosome. The spliceosome is a special enzyme that is made of a combination of small nuclear RNA, which is actually a catalyst that actually is made of a special type of RNA polymerase, and a special gene within your genome makes this small nuclear RNA that will serve as a catalyst in this enzyme. And also with small nuclear particles or proteins which gather together with the small nuclear RNA to create the spliceosome. So you see, those SNRPs is what we call them. They will get together to form a spliceosome. Now, the cool thing about the spliceosome is that different spliceosomes are made of different SNRPs. And depending on which SNRPs you have, you're actually going to cut different kinds of introns. And so maybe spliceosome type A will cut every single intron there is. But spliceosome type B will leave some. Spliceosome type C will leave some others, and so forth. And so depending on which proteins you are adding to that molecule, the spliceosome, you're going to recognize different boundary sequences, and different introns are going to be removed from the code. We'll talk about that, why that's important in a second. By the way, here's a suggestion of yet another role for the RNA molecule. It's a new kind of RNA we haven't heard about before, the snRNA, or the small nuclear RNA. It is part of the spliceosome molecule, and it has a catalytic role. In fact, there's yet a fifth role for RNA, Some RNA molecules act like enzymes by themselves. We call those ribozymes, and they show up all over the cytoplasm of the cell. RNAs acting like proteins. This is going to be important later in the end of the lecture series when we talk about the importance of RNA for the cell. And realize then that we now have two more types of RNAs. RNAs that act as catalysts within the SNRPs, and RNAs which actually act as enzymes in the middle of the cytoplasm. We call those ribozymes. Either way, what the SNRPs will do is they will get together to form spliceosomes, which will then recognize the introns, call them up, stitch them out, and basically remove the introns from the code. And then DNA ligase, the same one that works in the replication sequence of DNA, will come in and stitch that messenger RNA together and kind of make sure it's now one continuous molecule. And so by doing this, you actually created a mature RNA molecule that has only what you want inside of it. Why would you go through this whole mess? Why not just put what you want there? That's what prokaryotes do. Prokaryotes don't have any of this nonsense. They don't have any junk in the coding sequence. The coding sequence is only what you want. They have no junk whatsoever. But eukaryotes adapted to have introns. Why would you do that? Well, there's several reasons. Think about it. The more junk you have, the more target practice you have for mutations. What do I mean by that? If you have an army of 500 people and you try to storm a castle, but you have holograms which make look like you have 5 million people, it's going to intimidate the castle warriors, and it's going to make it harder for you to be hit as a warrior because they have about millions of fake targets. If the introns are there, mutations happening in the introns will not cause problems for the DNA because they're junk anyways. And that means that having introns in the DNA reduces the chances of mutations actually happening in the actual areas that matter, that actually code for proteins. So I think of it as mutation protection. Another reason for this is the idea of crossing over stability. Remember from the chromosome inheritance video that the greater the distance between two areas of the chromosome, the greater the chance that those areas are going to be crossed over together. In other words, if two genes are really close to each other, it's going to increase the chances that one piece will cross over while the other one will not. But that means that leaving those introns there enhances the chances that the gene will stay intact during a crossing over event because you're adding distance between them. It's kind of like a fancy way of solving the crossing over problem. Because even though crossing over was created to generate variety, what if you want to preserve the structure of the gene? Then that means that you don't want genes to cross over, and adding distance between the code actually enhances the chances of they sticking together during a crossing over event. Also, since you have different kinds of SNRPs and different kinds of spliceosomes, you're actually going to have different kinds of introns being removed. In other words, sometimes when you make the protein, you're going to leave every intron is going to be removed. But at other times, you might leave an intron there, and that makes a different protein. So that means that by cutting different introns out, you can make different proteins out of the same gene. When you actually look at the human genome, it doesn't make sense for there to be as many proteins as there are. The majority of the human genome, as much as 95% of it, is junk. So how can we have so many different kinds of proteins if we only have 5% of our DNA actually coding for proteins? The reason is, you can get the same genes and change them into different proteins by leaving or removing different introns. So it's kind of like you get a meal, and depending on which ingredients you put or take out, you make a different kind of meal. And so that's another reason yet why introns are there. Or in other words, differential RNA process adds more to introns, and that's why they exist. Also, because you have introns, you can add things to the proteins. Maybe if you leave an intron there, it might add a special domain or another function to the protein that wouldn't be there if you were to remove that intron. So there you go. That's how you make different proteins from the same gene, but depending on which introns you leave or take out. In other cases, the protein is not made at all unless all the introns are removed. In other words, this is a way that you can actually control whether or not the protein is made. Another way to do gene expression control. If you destroy all the spliceosomes which are involved in actually removing these introns, this protein will never be made. Because without the spliceosomes, the introns don't get removed, and then you don't have the protein. So you can regulate whether or not the protein is being made by making sure that the spliceosomes, which take out the introns, are not around. For all of these reasons, to protect us from mutation, for maintaining stability during crossing over events, to create different kinds of proteins with different kinds of domains, and to control gene expression, introns are part of the human genome and of a lot of eukaryotic genomes. Another change is also made to the RNA before it leaves the nucleus. And this is actually something that's coded by inside of the actual coding sequence or the transcription unit. You add what is called a GCAP. And the GCAP is basically a nucleoside triphosphate that's made of guanine. So you have whatever RNA message you have, and to the top of it, you write a GTP, or guanine with 3 phosphate groups attached to it. That is going to serve as the energy that's going to be necessary to initiate the translation process. And although it is possible to do translation without it, this actually makes translation process easier to jumpstart. And so that's what the GCAP is for. And it also protects the beginning of the message from degradation. And remember that after that, you have some junk, non-coding DNA before you actually get to the start codon. Then you also get the actual coding sequence, which is just introns. Then you have the untranslated region after the stop codon, which also helps to preserve the message. And then you do one last change before you get out of the nucleus, which is add a poly-A tail. At the 3 end of the molecule, you add a bunch of adenines. Adenine, all over again. And the bigger the adenine tail is, the longer this RNA is going to survive in the cytoplasm when it's trying to be destroyed by enzymes which actually try to destroy the RNA to control the gene expression. And so the poly-A tail, it's actually there to preserve the message and try to preserve that RNA long enough so that the protein is actually made. Remember, you have to have a way to destroy the messenger RNA, otherwise the protein will be made perpetually. In other words, at the same time that you make the RNA, enzymes are walking around trying to destroy it. So that you always need to be continuously making this messenger RNA if you continuously want to make the protein, because enzymes are constantly trying to destroy it. And that's a way that the cell regulates how long the actual action takes place. So you receive the signal from the cell, and the transcription factors activate the transcription. You make the RNA, and the RNA gets destroyed almost immediately if it gets caught by one of those enzymes. But if you have a long poly-A tail, it will actually last longer, and the protein synthesis would actually take place for a longer period of time. And that's what it's for. So to review, a small molecule called SNRPs, which is made of a combination of proteins and small nuclear RNA, which acts as a catalyst, and other proteins get together to form a spliceosome. Each spliceosome is different from the other, and it can cut different introns, which leads to differential RNA processing. But the point of the message is you're going to be removing those introns, cutting them out to make sure that the final RNA only includes the exons. Then you're going to add a poly-A tail and a GCAP head to make sure this RNA is ready, and you're going to have something that looks like this. And the RNA processing is completed. And after RNA process completed, you have a messenger RNA that kind of looks like this. A GCAP, an untranslated region to protect the message, the actual start codon, and all the codons in the sequence of the protein that you have. Then you have the stop codon, a bunch of junk again to make sure that the DNA doesn't get destroyed, and then the poly-A tail, which helps protect the message from degradation. And that's how the messenger RNA actually looks like when it's ready to do its job. By the way, within the downstream junk, you actually have something that's called polyadenylated signal, and that's how the cell knows where to add the poly-A tail. It's going to have to be a little bit after that shows up. And that's what that's for. After the RNA processing is completed, the RNA will exit through the nuclear pores, which are big enough just to allow things like transcription factors and the RNA molecule to exit, and the strand of the RNA will go into the cytoplasm and immediately start looking for a ribosome.